Hi, it's a pleasure to be here. I'm very honored to be part of this event today, and I know I'm probably more excited than anybody in the room. Normally I give talks to a lot of people my age and older who know all this stuff, but it's much funner to give a talk to people who are just coming into science and want to know more about science and are really have their whole life ahead of them. So I'm honored to be here and be part of this, so thank you. And so I want to tell you, I am a research microbiologist. I worked for the U.S. government for 18 years at the Food and Drug Administration. And one of the things that I'd like to tell you is that when I got my training in microbiology, I did it in a kind of a diverse pathway, which is kind of fun, which also shows you that you don't really need to know exactly where you're going to land until you land there. So for me, I started out in graduate school working on HIV and dangerous human viruses at the National Institutes of Health. And after that, I switched over to the Department of Agriculture, and I worked on bacterial diseases that kill plants, apple and pear trees. And then I became a professor at Loyola University in Chicago and started working on diseases of animals in Lake Michigan, like fish and things like that. So I've had a very diverse career. And so now I work for the Food and Drug Administration, and my job is to try to keep food safe from being contaminated with germs that can make us sick or even worse. And that's a job that keeps me busy full time. So I think I ended up at the FDA. It was almost fate that I come there, because there were three things that happened at the same time that brought me to FDA. It turned out my father-in-law hated the fact that I lived in Chicago and he lived here, because he never got to see his grandson. So it turns out that he taught students whose parents worked at the FDA, and he was getting me job interviews at the FDA every day. The second thing that happened was I actually applied for a position at the Food and Drug Administration, the way normal people do, and then at the same time I actually met a scientist who directed a lab at the FDA while I was at a big trade meeting actually here in Washington in this building a long time ago. So those three things happened at the same time, and as fate would have it, the FDA called me back, and I've been there ever since. But today I'm going to tell you something about the cutting edge technologies that we've used to actually help keep the food supply safer. We're using GENOMICS. You guys know about the Human Genome Project and DNA sequencing, and how we're using that to help safeguard the food supply. I wish that I was up here to tell you that we finally cracked the 5-second rule. And that in fact, I wish I could tell you that it is true, that yes, as long as you get it off the ground within 5 seconds, you're good. But that's not the case. There is no scientific data to support that. But I am here to tell you something else exciting. And it starts with the mission of the U.S. Food and Drug Administration, which is to provide you with a safe, wholesome, and sanitary food supply. And it seems very simple when you put it in 4 words, but I assure you it's not very simple. It's quite a challenge, and it's very exciting along the way. This is a summary in a nutshell of the problem that we're facing. It's about a minute and a half.
Food. So enjoyable, so delicious, such a satisfying and essential part of our lives. And yet, do most of us ever wonder where it comes from? Where it's grown? How it's handled and packaged and shipped? Nope. Most of us don't think about any of that. Not until something we've eaten makes us sick. And then, how do we figure out which particular food was the culprit? Think about a salad bar, where you might easily eat 50 different ingredients. Each different kind of food can come from a different part of the world, and each one has different pathogens associated with it. And that means that every time you enjoy a salad, you're really exposing yourself to a large number of global pathogens, any of which have the potential to make you sick. According to the Centers for Disease Control, foodborne disease outbreaks are responsible for about 48 million illnesses, 325,000 hospitalizations, and 3,000 deaths in the United States every year. As the world becomes more interconnected, maintaining a safe food supply has become an increasing challenge. And the Food and Drug Administration has pioneered an innovative response to this challenge. Collaborating with federal and state public health labs, FDA has built a pathogen identification network called GenomeTrakr. It uses whole genome sequencing to look at the DNA fingerprint left behind by disease-causing bacteria, allowing FDA to identify the source of an outbreak with more detail and clarity than ever before.
So that's in a nutshell what we're doing. It's a very important project. I would say that it may not be quite as important as the early work of some of the great microbiologists like Pasteur, Leeuwenhoek, Reddy, and Fleming, but it's very important, and it's very applied to modern problems in food safety. And so we are very hopeful that it will be a lasting solution to a long-term problem. Just to give you a little more about how complicated the food supply is, when you're trying to trace back an illness from how they got sick, what they ate, you can see that the FDA has a real challenge on their hands. There are 200,000 registered food facilities in our country, 82,000 here, 115,000 which import food on just about every day. There are 300 ports of entry that food come into the country every day, and 130,000 importers bringing that food in, and with 11 million lines coming in every year. And just so you know, an import line is actually a convoy of large cargo ships full of food. And of course, let's not forget there's more than 2 million farms in the United States that are growing food and putting food in the food supply for us every day. So that's a daunting task, and there certainly aren't enough inspectors in the country to help go around and check every one of those items as they come in. And of course, as the nice actor said on the screen, in our country alone, we suffer from about 10 million episodes of foodborne illness every year. 60,000 go into the hospital, more than 1,300 die, and Salmonella leads the way. It causes 1 in 10 foodborne illnesses every year. So I work on Salmonella. Salmonella is also a real problem because it's no longer just in chicken and eggs. See, when I was growing up, everybody said, just cook your chicken. Make sure your chicken's well done. You'll be okay. Make sure you never eat a runny egg. It's not that way anymore. Salmonella has invaded the ecosystem, and it's now coming out in products that we never dreamed it could even live in. So this has been eye-opening for us as well. And for us, the idea is to get the contamination out of the food supply as quickly as we can, and because that equates directly to saving human life. And that's what we're trying to do. So we start with a finished product, and sometimes it takes us through factories and facilities. Sometimes it takes us through farms and into the ecosystem where wildlife can also spread contamination. Sometimes it takes us overseas. It can take us anywhere, and it takes time. And so we're looking for tools, technology, to help shorten this time span and ease this burden. And so along came the whole genome project. And you guys know about the Human Genome Project. It started in the mid-90s. It cost about $4 billion at the end, but by the time it was all done, we knew how to sequence the human genome quickly and accurately. And we took that technology and we applied it to foodborne illness, bacteria, and it is doing exactly for us what it did for the Human Genome Project 10 years ago. In fact, many in the world, including Jorgen Schlund, who directed at the World Health Organization and now at the GMI, says whole genome sequencing is the biggest thing to happen to food microbiology since Louis Pasteur showed us how to grow them. And so it's a big deal. I like to say that maybe in the field of astronomy, it's equivalent to going from a backyard telescope that you bought at Costco to flipping on the switch that runs the Hubble. And that is no less what it's been for us. It's made that kind of impact. And so it leads us into the forensics of bacterial pathogens. And the good news is that getting the genome sequence is easy. It's just A's, C's, G's, and T's. So it doesn't matter what the pathogen is. One sequencing reaction will do it all. It's immediately useful, and all we need to know is a little bit about where the food came from and to have its DNA, the pathogen DNA, and we can trace it back. And so we use it routinely at the FDA over the last few years. And I'm happy to report that because of it, we're intervening much earlier, and we're helping solve foodborne illness with our federal collaborators much more quickly. Really, the way you look at a whole genome sequence tree is no different than you think of a family tree. It just shows you what bacterial pathogens are related to which pathogens. So using this sort of family tree, like in the Game of Thrones here, you can actually trace it back to its source. We should have known this a long time ago. Charles Darwin tried to tell us this in 1850. When he said, it's obvious whereby the occasional means of transport and continuous to land from America to Cape Verde Island, that such colonists would be liable to modification. And the point is that the genetics and the traits of every living thing will always betray their birthplace. And that is something that Charles Darwin knew, and all of us knew since, but he didn't have whole genome sequencing, and we didn't have it for a long time. So we couldn't look at those traits the way we can now. And now we can discern those birthplaces. I'm going to tell you very quickly, this was the test case. This was the example in 2009 that convinced us this was the way to go. And it happened from a spiced salami in New England. And it turned out that the salami was causing people to get salmonellosis. And it was a very fancy salami. So inside of it were pistachios and red pepper and black pepper and cheese and all kinds of good things that I like to eat over the holidays myself. So as it turns out, they couldn't figure out where the salmonella came from because every one of those raw ingredients had caused a salmonella outbreak with that exact strain the year before. And so it was a great mystery. And they came into the lab and they said, we know you just got this whole genome sequencing technique, but maybe you ought to give it a try now. I know we didn't give you much time to get it set up, but let's see if it makes a difference. And when we did it, we were able to go far beyond what was available here. This was the current technology. The old technology that we'd been using, they were all the same. We couldn't differentiate them using DNA fingerprinting like this. But when we did whole genome sequencing and sequenced all of their DNA, we found out right away that the pistachios were innocent, the cheese was innocent. It turned out it was the pepper. The red and black pepper, as you can see here, were completely exactly the same across their entire genome as those that made people sick. And so it very clearly pointed us to the red and black pepper as being the culprit for this outbreak. And so when we saw this, we knew we had a lot of power in our hand now, and we knew we had a capability that we never had previously. And we've been using it ever since. We can answer questions. Have we seen this strain or this isolate, this variety of strain before? We can compare it very quickly. Do these isolates form a cluster? Are they related to the illness? And then the big question is, can we link it back to people who got sick? And if so, then we can find the source and get it out of the food supply. Think about that salad that the actor just talked about. The salad has ingredients from everywhere. What a powerful tool to be able to go in to somebody who ate a salad and got sick and say, no problem, we now know it was the mushrooms and it came from some country far away. So we have that ability, and that's making a big difference. The people who make, grow, and process food can also use this technology. And they're using it now to determine whether they have real problems in their facilities or whether they have just a pass-through problem where something came from a farm or some raw ingredient and it's just working its way through the system and they did nothing wrong. And so this technology can help companies actually grow and prepare safer food before the FDA even gets involved in a foodborne illness. And so once we realized we had this power, we immediately wanted to share it with everybody, with the country, with the world, and we set up a big database and a big network called the GenomeTrakr. And we spelled it wrong intentionally so that when you Google it, you'll get our GenomeTrakr and not the 5 other GenomeTrakr's that I don't know anything about. So basically the idea is simple. We've put networks, sequencers all over the country in public health labs and universities, and everybody starts sequencing. And the key here is they have to share their data and they have to put it in a common database where everybody can benefit. And that's what's happening. And now because of that, public health practitioners, doctors, and companies and universities can all go back in and use the data for anything they need to answer real public health questions. Now, of course, all this costs money, right? So it costs money to actually put out sequencers, it costs money to have a network and all these things. But if you partner with the right people like we did at NIH right here in Washington, that part of it has been free because the National Institutes of Health stores genomic data for free. It's what they do. That gave us a lot more money to put a lot more sequencers around the country. So that's what we did. And the network is growing. There's a map of the network. It's both domestic and foreign. We have almost 50 partners already since 2013. But the other value that I want to share with you about whole genome sequencing is that whole genome sequencing doesn't just tell you where a contamination event may have started or came from. It can take the place of each one of these microbiological tests. Each one of these tests you see here costs a lot of money. And they have to be done on every germ that makes people sick from the food supply. But now, with this whole genome in our hands, it now can take the place if almost of all of these other tests. So we can learn just from that sequence what we used to have to do when we injected a mouse to actually see if the mouse got sick or how the mouse got sick. All of these things can be learned from the sequence itself. And that's another part of the revolutionary leap has been one microbiological workflow. And I like to show this slide because right away, the genomes are telling us things about salmonella that we had no idea. We had no idea that salmonella had evolved to persist and live in tuna and other top predators in the sea while it's waiting to get into its primary target, which is us. And so this is an adaptation that was new that we didn't know about. So it can live in tuna for quite a long time and not have a problem. So for us, for bacteria and foodborne outbreaks, the FDA has 3 steps that we generally follow. First, we have to find out, is there a problem? Is there a germ causing illness? We call that detection. The next thing we do is we have to do identification. And so we have to find out, what kind of germ is it? Is it E. coli? Is it Salmonella? Is it Listeria? Is it a virus? We don't know. So we have to find out what it is. And then lastly, we have to trace it back to where it came from, or else we can't stop future illnesses from happening. And so these are the 3 steps of a foodborne outbreak, or the anatomy of the investigation that we do. And right now, this new technology called next generation sequencing is taking over the back 2 steps, or these last 2 boxes of the investigation. And soon, when we can learn how to do the sequencing directly from the food itself and not have to put the bacteria on a Petri plate first, once we do that, it will take over all 3 arms of the investigation, and it will truly be one microbiological workflow. So in the future, scientists like you, when you become microbiologists and you walk into the lab, you won't pick up a pipetter and work with a flask or a beaker like we did, eons ago when I was young. What you'll do is you'll learn how to do genomics, and you'll actually learn computational sciences and lots of other things. It will be almost application driven. So when there's a salmonella outbreak, my boss likes to say he wants to have all this on his phone so he can pull it out and say, was it salmonella? Was it dangerous? What kind of salmonella was it? Was it resistant to any antibiotics that we used to treat it? Did it cause the outbreak, and what part of the world is it from? And does it have any weird adaptations that makes it hard for us to treat? And so these are the kinds of things we're going to be able to learn and have literally at push-button disposal in our hands using our smartphone devices. For the future in careers, it's just never ending, but 3 that are already happening in real life that we're actually hiring in the laboratory right now are what we call research genome microbiologists, who are microbiologists who actually learn genomics while they're in graduate school. We are hiring what are called bioinformaticists or bioinformatic traceback specialists, people who know how to write the programs and the computer programs that take sequence data and analyze it for us. And then finally, we're training people and hiring people who are coming out called comparative microbial genomicists, who can make sense, like I showed you that family tree from the Game of Thrones, who can build those trees and actually interpret them and advise the public health officials what their next steps should be. And so these are kinds of new careers that have spawned directly out of the genome program that's been developed in food safety. Some of the leaders already in that is David Lippman, who directs the National Center for Biotechnology Information, NIH. He and his team, including Jim Mustel there, his deputy and partner, are developing tools already that everybody, including you and I, can sit down at our computer at home or in the workplace and actually pull these data down and look for ourselves to see and learn what we want to learn. And so these are the tools for the future. They will be at your fingertips, and this is kind of the new microbiology. This is the way we want to move forward. We don't want to be in an ivory tower anymore. We don't want to have a closed system. We don't want to be strange to anybody. We want all the data in your hands. We want the tools in your hands so you can grow up feeling it and come into the field feeling it, for sure. So, and when you do genomics, it's a true village effort. Nobody does science by themselves anymore. And so we all work together, and the FDA works with the CDC and the USDA and public health state laboratories from all around and partners from around the world, including the World Health Organization. And so it's a never-ending collaboration of colleagues and friends that are all interested in one thing, and that is keeping food safe for the world. And so this is a picture of Mark Allard, who's my colleague who works with me closely, and my son, Ethan, who did a summer internship with us because he was learning genomics and is now in college studying it. So I think I may have timed that just perfectly. So I'm actually going to finish up by saying I'm happy to spend a few minutes entertaining any questions that you might have for me. All I ask is that you please come to the microphone so everybody can hear your question. Thank you.